Welcome to this BioinfoBytes explainer. Today we're jumping right into metagenomics, the invisible universe revealed. We're going on a really fast-paced, dynamic journey to uncover the hidden microbial ecosystems that are literally all around us. We usually define our world by what we can clearly see, right? Plants, animals, maybe the occasional single-celled organism if we've got a standard microscope handy. But right beneath our noses, and honestly right inside our bodies, exists this cinematic, impossibly complex universe of genetic information that we are only just now starting to decode.
All right, let's kick things off by completely shattering an illusion. For over a century, traditional microbiology has actually had a massive, glaring blind spot. It's staggering, really, but over 99% of bacteria and archaea are completely unculturable in a laboratory setting. For decades, if we wanted to study a microbe, we basically had to isolate it, stick it in a petri dish with some nutrient broth, and just cross our fingers and hope it multiplied. But the vast majority of microbes have incredibly specific ecological niches. They might need extreme temperatures, super unique trace minerals, or complex symbiotic relationships with other microbes just to survive. When we try to force them into a sterile lab, they simply refuse to grow.
Just imagine zooming in on a single teaspoon of ocean water, or taking a basic scoop of dirt. That tiny, everyday sample holds a massive, hidden microbial universe. We're talking about over 1 million individual microbes, representing thousands of interacting species. Because we couldn't culture them, we were functionally blind to the true complexity of nature. We were essentially trying to understand the majesty of the entire Amazon rainforest by only studying the 2 or 3 houseplants we could manage to keep alive in our living room.
So enter metagenomics. This is our brand new macroscopic lens. Metagenomics is the functional and sequence-based analysis of microorganisms extracted directly from an environmental sample. And the absolutely crucial point here is that it bypasses the petri dish entirely. Instead of trying to coax these incredibly finicky organisms to grow in a lab, we just go straight to the source. We take that teaspoon of ocean water, literally break open every single cell inside it, and extract the DNA directly from the environment itself.
This is a total game changer. Traditional microbiology is culture dependent, which, again, is kind of like interviewing one single person at a time to try and understand the vibe of a whole city. Metagenomics, on the flip side, is culture independent. By extracting the DNA of the entire community all at once, we simultaneously capture the genetic data of all the organisms in an environment in one fell swoop. We aren't limited by what we can grow anymore. We're now only limited by our ability to sequence and compute the raw data.
Okay, so how exactly do we read all this environmental DNA? We basically use 2 main sequencing lenses. First up is 16S amplicon sequencing, which acts as our taxonomic roll call. It matches specific, highly conserved genes against extensive libraries to answer one simple question: who is there? But if we want the full high-def picture, we use whole genome shotgun sequencing. By shearing and sequencing the entire genomic DNA from the sample, we captured the whole shebang. Not just the taxonomic markers, but all the functional genes too. So shotgun sequencing tells us both who is there and what are they doing.
Now, as you can probably guess, extracting all this DNA creates massive, chaotic streams of raw data. To actually make sense of it, the data has to pass through a strict, high-speed bioinformatics pipeline. First, those raw reads are run through vital quality checks using tools like FastQC. That's followed very closely by host read removal, which is absolutely essential, so we're only looking at the microbes, not the host's DNA. Next, we use taxonomic profiling with tools like Kraken2 or Metaflan, mapping millions of short sequences to tell us exactly who is present. Finally, functional profiling with human N jumps in to figure out what those genes actually do.
And what happens next is basically magic. All that kinetic data processing instantly organizes this chaotic, invisible genetic soup into beautifully structured phylogenetic diversity maps. We can literally see the deep branches covering extremophilic microbes, especially across the archaea domain, along with the vast networks of bacteria and eukaryotes. We can now map the exact architecture of microbial ecosystems, plotting out complex evolutionary relationships that were completely invisible to us just a few decades ago.
But let's ground all this high-tech machinery in the real world, because the applications are wild. Take the human gut microbiome, for example. Collectively, the microbes hanging out in your gut right now contain 100 times as many genes as your own human genome. We're also using this tech to survey massive ocean microbiomes and plankton networks. And crucially, we're using it for antibiotic resistance discovery, finding novel mechanisms hidden deep within environmental samples, unlocking solutions that traditional methods would never ever catch. So what is the ultimate takeaway here? It's that one sample holds an entire ecosystem. We have officially moved from the microscopic to the macroscopic. Metagenomics provides this incredible continuous stream of data unlocking the secrets of our world, proving that a single drop can reveal an entire universe. We no longer have to isolate life to understand it, we can read the blueprint of life exactly where it lives. So what unseen universe might be hiding in the soil of your own backyard, just waiting to be sequenced? Keep questioning, keep exploring, and thank you so much for joining me on this explainer.